Hi, everyone. My name's Sebastian. I'm a degreeless heathen working out of a home lab in Huntington, New York, and I'm the host of Tears and Rain. And for today's podcast, this is episode number 6, and we have Lee. So please introduce yourself and tell us your story.
Hi, I'm Lee. I'm an assistant research scientist at University of Georgia. I study plants. I study plant specialized metabolism, and I switch field, which I'll explain that in a bit. So, and Sebastian, it has been great to be on here. I would like to thank you for this podcast. It has been a great project. I watched um, every one of the 5 episodes up to now. So I guess the first question is how I get into um, graduate school or what got me on the path.
Yeah.
And it's going to be a little bit of a long answer because I don't know where to start. I guess I'll start as a kid, right? So I think that I don't know how to explain this, but ever since I'm a kid, I think I just had a thing for um, nature. I remember when I was a kid, I had this really thick book of children's encyclopedia,
Mhm.
and of course it's in Chinese and all that. And it's a colored and picture book, and and I loved that book as a kid.
Hmm.
And I would spend hours and hours um, going through each page of the book. And the pages that I spend the most time on are those related to plants, animals, or different subfields of natural chemistry, stuff like that. So I knew that as a child, I would love to do this if I
Hmm.
get a job in a uh, science related field. But it's unclear what I would study until many years later. So I like plants, right? And one of the places that I would like to go as a kid is so I was born and raised in this town called Guangzhou, China, and it's a big town with I think I want to say 10 billion people, 10M, and it has a big botanical garden, uh, the South China Botanical Garden, and I spent all the time in that botanical garden. It's a big one too. It has hundreds and hundreds of acres of just greenhouses and gardens. And I think I like plants, not in the sense that I like the flowers. I do like the flowers, but I just like how green and how green the plants are,
Hmm.
and how diverse the shapes and forms are. Yeah, so that's the things that I enjoy the most as a kid. And in terms of what really gets me into graduate school or research, I think I can count back or recall back from high school. I remember one day we had this high school class, and it was a really brief introduction to plasmid, I believe.
Hmm.
It's this idea that you can take a gene from one species and you can insert that into a plasmid, and then you can put that plasmid into another species, and then it will work as if it's its own gene. And that was how it was introduced, and it was a really, really brief introduction. It's high school, right?
Mm-hmm.
And somehow that just shocked me. I was like, wow, magic. I need to learn how to do this. And I need to learn how to do this with my own hands. So that's the thought that I had
Cool.
in my head. So I was saying, okay, I need to go get a college degree in a place that I can do undergraduate research, ideally on plants because I like plants. So that means I want to do recombinant DNA research in a plant lab as an undergrad. And I thought, what would be the best place to do this? And I was told that if I apply to a research intensive R1 university in the United States, there's a good chance that I will be able to work in the plant lab and do that kind of research that I want to do. So that's how I came to US. So I applied to a bunch of colleges and schools, and I was very lucky to have got into UC Davis. And then at that time, I chose biochem as my major. I wasn't sure which major to pick, so I just picked biochem because it sounds about right. And that's where, that's essentially how I get into research and how I came to the United States. So then, I think it's not until the third year I joined a plant lab. Join a lab. So I think in year number 2, I took intro biology, and then one of the professors studies plants. So then after I got an A on the class, I asked him, hey, can I work in your lab? I got an A in your class, so that's good enough. And then Kay was like, yeah, sure, you can come work in my lab. So that's basically how I got started.
What year was it when you got into that lab? Just to frame a picture.
Yeah, so I am starting my third year in college
Gotcha.
when I started in the lab, and that was 2014.
2014. Did you already have kind of in the back of your mind a topic for your project, or was that steered by external factors or your advisor at the time? How did you come up
Yeah,
with your topic?
well, so that was completely, completely coincidental. So, so I basically said I want to do recombinant DNA research in plants. And Kate was like, "Great, here's a project, go for it." So at that time, we were looking at, well, it's a long time ago. So let me try to recall what the project is. The lab was looking at stress response from tomato plants, especially the earliest stress response, like just a couple days after the stress hit. So they had this big RNA-seq data set of a stress time course, and they have found genes that are quickly induced at the earliest stage. And they want to clone the promoter of those early inducible genes and use them to drive a fluorescent protein. And the idea is that if we use those early stress inducible promoters, the fluorescent protein will be expressed in the earliest stage of the stress, right?
Sure.
So then I was asked to, Lee, can you help us build this control plasmid first? So I said, okay, sure. So then I learned how to, so I basically clone the chlorophyll AB binding protein, which is always expressed, and it's only expressed in basophil cells, for example. So then I cloned that and I fused that with a green fluorescent protein, assembled it into a tDNA plasmid. I learned how to transfer it into agrobacterium, and then I transiently express it in Banff, which is the same skill that I still use now. And that's really amazing. And then 2 days after the infiltration, I brought it to the fluorescent scope to see if it worked. And I was really nervous because I don't know if it's gonna work. Mm-hmm. Everything is so fresh, I did what the protocol asked for, but I just don't know if it worked. Mm-hmm. So then I go to the scope, and then I cut off a small piece of the leaf and load it on the scope. Then I looked. I was shocked. I see this bright green fluorescence protein, but not everywhere in the leaf. It's only in the basophil of the leaf, but not in, say, the vasculature of the leaf. And I was shook because wow, this actually worked the way we think it did. Mm-hmm. Not only we're assembling 3 species in one thing, right? We took the Arabidopsis promoter, fuse it with a jellyfish gene, shove it in agrobacterium, and then that goes into a tobacco plant. 4 species, and even after that, the thing worked the way we intended, and I was shocked. And I know that this is what I want. I want to do this for my career, and I want to teach others to do this because this is so fun. This is great. Mm-hmm. So that's what made me consider doing a PhD, because after that I just can't stop doing that kind of experiment.
That's so relatable in so many ways. Yeah, like that first initial proof that this is actually real, and that things are actually working, but with your own hands, I think is so important. So yeah, I deeply resonate with that. So tell us in terms of like the topic, because that always is a big question of how do you find your topic? How did you get to the point where you decided on pitching the idea to your potential committee?
Yeah, so I'll just go through how that worked for my PhD program. So then in year 4, I applied to a bunch of plant biology PhD programs, and then after that I just stayed at Davis to do a PhD in plant biology. And so we don't start with a PhD project. So each student can do 4 rotations. So they basically pick 4 labs, and they spend, I think it's 5 weeks in each lab. And after that, the lab and the students would try to align, right? Mm-hmm. So at that time, I picked 4 labs. All of them have some molecular biology component to it, because that's what I like. And they all have some kind of crop component. For example, I rotated in a soybean lab, a rice lab, and a barley lab, and a pomegranate lab. So that's the 4 labs. And at the end, it comes down to the environment of the lab, as well as which lab is looking for a student, basically. So at that time, so I joined my PhD lab, right? And at that time, so the lab was just awarded a grant, a NSF grant to study the small RNA in rice reproductive cells. So the PhD advisor was trying to hire a new PhD student to work on the grant. So he needs a student, I need a lab. So that is how I got in the lab. So I asked myself, am I interested in this project? And the answer was yes. So it has to do with plant reproduction. The project is like small RNA in rice egg cell, sperm cell, and zygotes, right? And I recall that I really enjoy the plant reproduction part of my plant development course that I took as a college student. And it was only a week on those topics, but that gave me a deep impression. So that's like, okay, I think I'm interested in this. I can do this. So that's how I started to work on small RNA and plant reproduction.
Gotcha. So could you give us like just a really brief, casual abstract of like the general project?
Yeah. So let me see, how should I start? Right. So I'll start with this, right? So we have the sperm cell and the egg cell, and they fuse to make the zygote. And the zygote will go into an embryo, which will go into the next generation. And this is true for both animals and plants. And one basic question, or one fact, is that the egg cell is 1,000 times larger than the sperm cell. And the egg cell actually contains most, if not the RNA, that is required for the early stage of the embryo. Then the question is, how come the egg cell just won't go ahead and start to make the embryo? Why does it have to wait for a sperm cell if it has most, if not all, the RNA that is required to make the embryo, right? So, and the lab was interested in studying why that is the case. And at that time, we cloned a gene called baby boom 1, which is expressed in the sperm cell. And if you express this gene, baby boom 1, in the egg cell will go ahead and make the embryo without the need for a sperm cell. And the lab was interested to understand how baby boom 1 is controlled or not expressed in the egg cell. And then one of the hypotheses is small RNA. And so in plants, there are 2 big categories of small RNA. One class is about 21 to 22 nucleotide in length, and those targets the mRNA, the transcript. They either causes the cleavage of the mRNA, or they stop their translation. So those are called post-translational gene silencing, or PTGS. And the other class is the 24 nucleotide siRNA. And then the 24 actually targets the gene itself and then targets DNA methylation. And those are transcriptional genes silencing. So we thought that either of these could be responsible for keeping the embryonic genes in check until the sperm cell arrives. So that was a hypothesis, and I thought it's a good one. And spoiler alert is not that. And we still don't know how, but that's definitely not how it works.
Interesting. I mean, it's a pretty sensible hypothesis, right? Interesting.
Except it's wrong. It's wrong.
Wow. And I mean, that's already a great lesson of just like, even though it's sensible and elegant and clean, doesn't mean it's always going to be right. Yeah. Wow. Okay. Yeah. So you have your project, and now you're going into your first year, right? So what was that like? Like, how was your interactions with your advisor? Like, tell us a little bit about that.
Oh, it was a very interesting experience. A little bit spicy. Okay. So I joined the lab, and one of the main reasons that I joined this lab is that I think, and I still think, that me and my PI will vibe. I think that's actually the most important thing. You know, when you do a rotation, I think the thing that you should check for is that if you can get along with your PI, one of the most important things. And my PhD advisor is very hands-off. And then he requires that the student need to have, you know, some degree of independence, which I have, and I thought it's gonna work, and it did work, but it's a very interesting process. And then so I started in the lab, and he was like, "Li, here's the thing. I don't want to train you, so you should go train yourself. And here is the copy of the grant. It has all the experiments, everything that you need to do. And now you go for it. Bye."
Wow.
And then if you have anything that you want to talk to me, please come talk to me. But otherwise, there you go. Here's all the reagents. Here's the grant. Here's everything you need to know. Go. And it's sort of a sink and swim situation. And I think most graduate students are not able to do this. And I was really glad that it worked out. But usually it won't work, but it worked. And that was kind of an interesting experience. I spent a lot of time just reading on my own, just, you know, try to understand everything that was in the grant. And then I read the instruction of all the kits, all the reagents, try to understand how they work. And yeah, so the first year was quite an experience.
Wow.
And I haven't, you know, done R or statistics or bioinformatics at all. So I also learned all those on my own from scratch. So the first year was an interesting experience. Okay, and then I guess I'll just explain what the chapters are. So the first step is, in order to profile the small RNAs in those reproductive cells, step one, you need to get those reproductive cells. You need to isolate those reproductive cells. And that was a pain in the butt, okay? And that was an understatement. Pain in the butt is an understatement. So we chose to study rice because for rice, you are able to dissect out the ovary from the flower, and you can cut open the ovary, and then you can gently squeeze on the ovary, and the egg cell will just pop out. And after that, you use a fine glass capillary to literally pick the egg cell out and transfer that to a tube.
Could you give us a sense of scale? Like how big are the things you're manipulating?
So the ovary is about that big, and you have to choose a dissection scope. And the egg cell, I want to say, is 15 micron across,
Wow.
which is relatively big for cells,
Sure.
so that you can see them quite well under a scope. And then even the 5x length, you can see the egg cell. And then you just need to use the fine glass capillary to aim the egg cell and pick the egg cell and transfer that into a tube. So I have to learn how to do it. And it took me, I want to say, 2 to 3 months to actually get a handle on this.
Wow. Wow.
And then it's just the grind to get enough egg cell so that we can get enough small RNA to make the RNA-seq library. And that was pretty much, I want to say, 6 to 8 months of year 2. So I sit there every day, and then I pick egg cell for, I want to say, 6 hours a day,
Oh my God.
and maybe 4 days a week.
Wow. That's a grind. That's like a monastic grind. Like just like laser focused. How did you maintain your focus throughout that? Is it just like a desire to push through, or like what did you dig to get to that point?
No, I think I'm just a very curious person. I just want to know
Okay.
what other small RNAs are there in those egg cells.
Sure, sure.
Because apparently no one else on the planet have done this. And I think I'm still the only one who did it because it was so hard. Nobody will.
Sure, sure. Is it like what's the abundance like of those microRNAs? Like, give us a sense of how much did you have to collect to just get one preppable amount?
Yeah, so the egg cell is relatively big, so it has a lot of RNA in it. So that means we don't need hundreds and hundreds. So at first we started with 30 egg cells. So then that gave enough RNA. And then we bought this kit, and then the kit was like, well, if you have 10 nanogram of total RNA, it will be enough to make a small RNA library. So then I did some experiments. I think 30 XLs will be safe, and that will be more than enough for the specification of the kit.
Mm-hmm.
But it still takes weeks and weeks to make one rep, and then we will need at least 3 replicates, right?
Right.
So it took months and months to do this, and it's easier in the spring up until, I want to say, the spring through August is great since the rice are happier and it's more warm, and then it's not as great when it gets cold. Even though the rice plant is in the greenhouse, they know.
Just tucking back in. Yeah. Okay, so you collected that, you made the RNA library, and did you immediately go for sequencing as the next step of your project?
Yeah. So then after that, that was also a very long and interesting process. So then I collected those egg cells. So basically, I have these tubes of egg cell that I store in the -80, which it took me 8 months to collect. And then I was like, okay, now it's time to crack open those egg cells to get the RNA out of it and make library. So that was towards the end of year 2, okay? And I was really scared. I said, what if I screw it up? I'll just waste all those efforts, all those months collecting the egg
Yeah,
cell.
it sounds terrifying.
So then I need to convince myself the kit is going to work. So then that means I need to make some practice material to test the kit so that I know the kit is going to work. So the first practice material is rice seedling because it's easy to get. So what I did is I ground up a bunch of young plants, and then I diluted the RNA to about 10 nanogram and use that to start with the kit. And that was immediately a problem.
Uh-oh.
So then that was on the low end of what the kit specified. And then after I went through the entire library prep process, the yield was too low to put on a Deluminar instrument. And then I was like, oh no, okay, I need to optimize this. So then I spent a lot of time trying to understand why the yield is too low. I talked to other labs who have done this, and at the end, I just emailed them the technical support of the kit. I said, I cannot get your kit to work.
Sure.
Here's what I did. I couldn't get your kit to work. And then 2 days later, they email me back and say, have you tried this? Have you tried that? I was like, great, I will go try this. So, and after that, a few more weeks later, I modified my protocol, and I could get the yield to an acceptable range for the instrument.
Still with rice seedlings, right? Not with your, yeah. Okay.
And then that included using less adapters and more ligation time in the adapter ligation step, as well as more PCR. Okay, so that was the first step. And then step number 2, so I was still unsure if it's going to work. So then step number 2 is I then make protoplast from the rice seedling, which is quite light, the egg cell. And then I counted exactly 30. And then I put those into a tube, and then I crack those open to make RNA. Then I go use the kit. And I was able to get it to work. So then using the same modifications and exactly 30 rice seedling protoplasts, I was able to make those library. And at that point, I was convinced that I can probably get this to work. Then I tried the Excel, and it worked. I was able to make those libraries and drop them off to the provider, and I was able to get those reads back. That was a nerve-wracking journey.
I can't imagine the pressure there. Was the yield just like what you expected, and everything just worked out in terms of processing, or was there any scary moment?
Yeah, so that was enough yield to be put on an instrument.
Okay.
Not the best libraries that you will make, but just good enough. And we're able to get reads back.
Gotcha. And now so now you're shifting over to analysis, and you said that you basically taught yourself R during that process, right? So how was it like being like a novice person at R, also being in charge of analyzing your own data with like not much guidance?
Yeah, so the good thing at that time is that we had a collaborator who is at UGA, and he had a lot of experience with small RNA analysis.
Cool.
So I basically just asked him, hey, what do you usually do with this? And then he was like, here are my 2 papers. We do this kind of analysis. I was like, oh, great, okay. Thanks.
Nice.
So that wasn't conceptually too hard.
Mhm.
It just I need to learn, R coding, bioinformatics, stuff like that.
Sure. Just a casual walk in the park, right?
Mm-hmm.
Yeah.
And then the well the difficult part of the story is continues. Okay,
Mm-hmm.
so we get the reads back. So and as control, I also sequence some seedling small RNA. Just as a control, I want to know if the seedling look like what we expect from, say, leaf, right?
Sure. Yeah. Makes
So the
sense.
good thing is that the seedling, even though it's like low RNA concentration or like small number of cells, they look exactly like what others have seen in rice leaf and rice fruit. Okay,
Yeah.
great. So it worked. But then the egg cell, that's the problem.
Oh no.
It's not like anything we have seen.
Hmm.
So it's completely not what you expected. And what happened is that in those egg cell small RNAs, those small RNA are concentrated at discrete sites of the genome, and they don't appear to match any genes or any transposons, which is not what you expect at all. Usually they either correspond to transposer elements right next to genes, or they correspond to genes, okay? And then those discrete sites in those XL small RNA, they are not either of those. And I was really concerned. I said, what does this mean? Does that mean what the good thing is that all three libraries look like each other?
Sure.
At least it's consistent.
Right.
Whatever it is, it's consistent. So at that time, we didn't know what it is. So then I was like, oh, maybe 30 cells is not enough. So then I went back, spent another half a year, collected another 3 reps of Excel, but now each rep has about 50 cells instead of 30 cells. So then I do the entire thing again, and it came out just like the first 3 replicates. So at that time, I was convinced that this is actually real, that this is what the Excel has.
Mm-hmm.
It has this distinct clusters of small RNA that does not seem to match any genes or transposon. And to convince myself, then I tried the small RNA from ovaries, which is the tissue outside of the egg cell. And it turns out that ovary has exactly the same pattern as the egg cell. So that then I know it's biological. So not just the egg cell, but actually the cells outside of the egg cell, they have the same pattern. So I was a little bit of a comfort. So it's not just an egg cell thing, but it's a ovary thing. Then I was convinced. Well, so I was convinced. For part of me was convinced, but then the other half of me was like, well, what if it's just contamination from ovary cells? So then I have to address that. And the experiment to address that was that I did the egg cell dissection, and then instead of isolating the egg cell, I just don't pick any cells. I just pick some cell-free juice that flow out from the ovary during the dissection process. So then this sounds like a really stupid experiment, but I actually did that. Then I cracked open those cell-free juice to make some RNA and to make some library out of it. Then I did qPCR to quantify how many molecules of library are there in the library that made from cell-free juice. And the good news is it's very, very few. So it's not contamination.
Cool.
And at that point, I'm finally convinced that this is what the XL has. It has some very strange small RNA profile that's not at all what we expected. That was a very long process. It took me, 1+ year, almost 2 years to be able to convince myself that what I got is sound.
Right. The contamination. I mean, that's a really elegant way to make sure that you're not crazy and like looking at the contamination. But did that idea come from within, from your own just kind of thinking, or was that?
Yeah.
Yeah. Okay. Wow. Because I'm just like looking back at some experiments that I wish I ran better controls for, and then like I know it now in hindsight that I should have done that or that. But I was always curious to know, like, what was the logic? I mean, what was the thought that got you to that point that you were willing to do something that, like you said, felt kind of silly, but was actually really important?
Yeah, I don't know. It's just like, well, I think I should do this, that kind of thing.
Mm-hmm.
It's like, because that's what, if there's contamination, it will have things come from the juice that flow out from the ovary. And I showed that in that juice, there's very few RNA in there.
Yeah, like I could see that like a reviewer number 2 question would be like, but did you sequence the juice? Right. Yeah, very cool. Okay, so now you're sure that this strange clustering is happening. What would you do next? What was the next logical step?
Yeah, so that was step that I was stuck there for a long time because I was at a loss of what this even mean. And for like a year and a half, I don't know what it means. And to be frank, I still don't know. But then, at that point, we decided to write a descriptive paper describing what the small RNA transcriptome is in the egg cell, in the context of seedling and ovary. And we also make the small RNA transcriptome for the sperm cell. And the sperm cell is also completely unexpected. The sperm cell is not like the egg cell, but it's also not like a normal cell. And to convince myself, I actually made 8 libraries of sperm cell, and they all look like each other. Then I was convinced that the sperm cell is also sound. But we don't know why the egg cell has a strange small RNA transcriptome, and we also don't know why the sperm cell has a strange small RNA transcriptome. And for a year and a half, I don't know what they do. I don't know what that means. And my PhD research was like, just write a descriptive paper. It's great. No one else has been able to do what you have done. And this is some, very unique data that the world should see. I said, okay. So then we wrote a descriptive paper describing the small RNA transcriptome of the sperm cell and the egg cell. And the good thing is that we know that our hypothesis cannot be right because the small RNA transcriptome is not what we expected. So that was chapter 2. The chapter 1 is just isolation of the reproductive cells. And then chapter 2 is the description of the small RNA in the egg cell and the sperm cell.
Gotcha. Okay, so you don't know so you've essentially invalidated your first hypothesis, right? With this, where did you go next? What was the next turn in the adventure?
Right. So at that time, I was trying to do 2 experiments. The first experiment is to sequence the small RNA in the pollen vegetative cell or the pollen tube cell to see if the small RNA in the sperm cell actually traffic or traveled from the pollen tube cell. And it was a hypothesis in the field, and people think that was the case, which explains why the sperm cell has, which will explain why the sperm cell has a strange small RNA transcriptome. And I could never get the experiment to work. So to get the pollen vegetative cell, you need to first break the pollen, because the sperm cell, the two sperm cells, are engulfed inside the pollen vegetative cell. So to get the pollen vegetative cell, you have to break the pollen first, get rid of the two sperm cells, and then you use the rest to make RNA. But because you have to break open the pollen cell, a lot of the RNA in that prep is totally degraded.
Sure.
So I was never able to get good quality RNA out of those pollen vegetative cell preps. Then I tried a few things. I tried to use a different kit. I tried to use homemade reagents to get good quality RNA out of those pollen vegetative cells. And I could never get good enough quality RNA to make those RNA-seq library. And then I asked the boss, I was like, hey, I've done this for 4 weeks now, and I could not get it to work. And he was like, well, I guess we can just give up and work on the next thing. And I was like, great, thank God. So I guess the lesson is that it's okay to shelve your project if you cannot get it to work.
Look, it's a lot better than throwing yourself against the wall over and over again, right?
Mm-hmm.
Okay, so there was a shelving point, and then
Mm-hmm.
I'm assuming a pivot, right?
Yeah. Then the other project is to get the small RNA from the zygote to see what happened after the sperm cell have met with the egg cell, and see how it have changed from the egg cell to the zygote. And for that was a much smoother process, because the zygote basically is the egg cell. It just has been fused with a sperm cell. So you isolate them the same way. It's just after you hand pollinate the flowers, you get it out like 9 hours after the 9 hours after the hand pollination. And then I make small RNA, and I get those reads back, and it's fine. So the thing is, the egg cell, the zygote look exactly like the egg cell. Well, not exactly, but in the grand scheme, the zygote look quite like the egg cell.
It's the same kind of distribution.
Mm-hmm.
Gotcha.
So the most abundant small RNA in the egg cell remains to be the most abundant in the zygote. And this means that a lot of those RNA are probably just carryover from the egg cell, the same RNA that was in the egg cell. And the most abundant small RNA in the sperm cell becomes very lowly abundant in the zygote. And you have to remember the sperm cell is really, really small compared to the egg cell.
Sure.
So that means the content of the sperm cell must have been diluted in the egg cell.
Sure.
And then the most abundant things in the sperm cell will become not abundant in the zygote. And the most interesting that came out of it is that then I compare what is new in the zygote versus the egg cell. And then it turns out that the new small RNA that was in the zygote are a lot more like what we see in the seedling. So this means that even at the most early stage, the earlier stage, the zygote is already initiating a reset from the weird profile of the egg cell back to what we should see. And that was interesting observation. So that was basically the main conclusion of the third chapter.
Gotcha.
So the third chapter was a lot easier because we already seen all the strange stuff. And we already seen signs of the zygote returning to the expected.
Yeah. It'd be a really interesting like time series experiment to see how the different RNAs decrease as it turns into more body cells. Cool. Okay. So you saw the weird stuff. And so chapter 3 was a little bit easier. Can you tell me what was the next step for the project beyond this point?
Yeah, so that was all 3 chapters. So by the time I completed chapter 3, I'm already in my year 6. So I was looking for jobs and all that. So I was able to complete the draft and the manuscript for chapter 6 for the third research chapter. And now I need to write the dissertation. So the writing part was actually not that hard because in my graduate group, you are allowed to basically staple your peer-reviewed article into one, and that becomes your dissertation. So what I need to do is I need to write a introduction chapter and a conclusion chapter. So that will make it 5 chapters instead of 3.
Gotcha.
So I have 3 chapters for research that I already completed. So I basically just need to staple all 3. And I wrote the introduction, which was a brief introduction. It's only 5 pages because each of the chapter have their own introduction. It's basically just an overview. And then I have the conclusion, and that was also, I completed it in 2 days, I think. It's basically say, here's what we find, and here's what we don't understand. And I also write a little bit about if I were to continue this line of research, what would I do? Then I propose that instead of small RNAs, we can look at chromatins and histone marks that are in those reproductive cells. And a few years later, the lab did get a grant to study that. So nice. that is going to happen.
Yeah, have a little bit of a steering towards it. That's cool. So as the ink was drying on your thesis, one question that I enjoy asking is, did you feel like you were the expert of this topic? I mean, you were probably one of the first, if the only person who's ever done that. But did you feel like you had enough to be able to confidently explain what you were observing, what little you could understand of it?
Yeah. So I think I am, and since I went through the entire process, I basically saw how, you know, and I make those libraries and I put a lot of work to convince myself that those are sound results. And I know the literature well enough to interpret why this is not what we expected. So I think yes.
Yeah. So during your defense, did you get how was it like defending a thesis that didn't have a fully resolved hypothesis in that sense? Like, what was some of the feedback you gave, and how did that pan out?
Yeah, so for defense at the Davis, you just give a talk, and there's not much, not many questions,
Hmm.
and obviously the question did come up like, why do you think the small RNA transcriptome in these cells are like this? And I said that there are, I can always speculate, right?
Sure.
So one of the speculation is that the small RNA in the egg cell and the sperm cells, they might target chromatin modifications, so histone modifications, instead of DNA methylation. And since we don't have the chromatin data on those cell types yet, so we don't know if that's true. But we have a grant to do this in my PhD lab now, so at least that hypothesis will be tested.
Gotcha.
And then the other hypothesis is they don't do anything. They actually don't do anything. They were just some sort of weird byproduct of the reprogramming of the floral organs, the ovaries and anthers. They were important in the floral organ, but they're actually not important in the gametes. So not everything have to be important. They just happen to be there, and it's not what you expected.
Yeah, that's a important lesson that evolution doesn't have to make sense to you, right? Very very cool. So so tell us a little bit about what you're doing now and how your experiences previously impacted that.
Right. So in my year 6, right? So I was applying for jobs, and then I applied for a bunch of postdoc positions. So I wanted to be a PI to have my own lab, and I think to me it's more rewarding to train a student to do an experiment rather than I go do these experiments on my own. So that was the main driving force to be a PI. So then I applied for a bunch of postdoc positions, and I interview at a few labs. And my current lab is the only one that worked out at the time. So I basically switched fields. So I switched from studying plant reproduction to studying plant specialized metabolism. And it's kind of a big shift, also kind of not a big shift, since I'm still using the same sort of experiments that I learned from when I was undergrad. and I still use the same kind of bioinformatic analysis that I use as a grad student. So it's actually not that big of a shift, and I think all the skills transferred to my new lab, which is great. And now I have both sets of expertise from both plant reproduction and plant specialized metabolism. So this is actually great.
Yeah, the pictures you post about the transient petal expression. I mean, I'm biased. I love flowers, but like, is that an established process in your specific sub niche? Yeah.
Yeah, yeah.
And about your model, oh, why is that one a target? Because normally people study like Arabidopsis or some Brassica or Props.
So for specialized metabolism, the good thing is that you get to work on strange plants that make strange metabolites, right? So the species that I study, Catharanthus, which makes two chemotherapeutic compounds, vinblazin and vincristine. Both of them are clinically used to treat various cancers, and they're very expensive. So when I started as a postdoc, the lab was interested in studying how does the plant make those compounds, as well as how does the plant control how much of those compounds it has made. So then we find new candidate genes, new candidate enzymes that are involved in the production of those compounds, as well as we find new candidate transcriptional regulators that turns on the pathway in the plant.
Cool. The if you one important kind of like hindsight question is, if you could go back in time and tell yourself a bit of advice, what would it be? Like, when during your journey would you have thought that a time traveler version of you to tell you the future would be great right now?
Well, there are a few things. One is hypotheses are not as important, and your hypothesis could be extremely wrong. Just don't be too obsessed with it. That's one thing. And two, the other thing is it's okay to shelve your project. If you can't get it to work, just move on. And you might come back if you find a better approach. I think we should be both persistent and agile. We should be we should keep trying stuff if it is promising, but we should be agile in the sense that if things are not promising, we should put it aside to move on to study things that is promising, right? And then the last one would be things are not as important as you think. I know that sounds strange, and that's more of advice to me, but not to others. Things are not as important as I think, as I thought. So, for example, at one point I would say that, well, I have to get an A in this class, I have to do this experiment by the end of the month, and it all seems so important. If I don't do this, it will be the end of the world. But no, in the grand scheme of things, in the span of 5 years, 10 years, it's actually not that important. So things are not as important as you think. It might be still very important, but it's not just as important, not as like war-ending important
Sure.
as you think.
Yeah. Now, when you were describing this, obviously it's through the lens of hindsight, but I noticed you had like a pretty stable amount of confidence from chapter to chapter. And for the folks who might feel a bit more insecure about their abilities, what would be your advice? How did you muster up that confidence to keep going and to be so independent? Yeah.
Well, friendship. I think having friends in grad school is extremely important, especially with your cohort. So I'm So the UC Davis plant biology program has a very cohesive student community, and we have happy hours every first Friday of the month. And and I have cohorts that work on the same floor as me, so that I would just go chat with them all the time. And from early on, I know that each of us have different expertise. There are stuff that I don't know that they know extremely well, and there are stuff that they don't know but I know quite well, right? So that's one thing that help build my confidence. And you should try to help them when you can, because that helps you as well. That helps you build your own confidence. And don't think, oh, if I help them, I don't have time. No, just go help them when you can, because when you help your friend, you help yourself. Help your own mind.
Mm-hmm.
So, having friends is extremely important. And be generous with your time with your friend. Because, again, helping your friends is helping your own mind. So that's one thing. And the other thing is training and teaching undergraduate students. Because when you are doing research in the lab, you are always comparing yourself to other people who have a PhD or who is going to have a PhD. And obviously, when we're at that stage, we are all, great researchers. But you don't remember how far you have come along. But then when you start to train a new undergraduate student, or you went to go teach an undergraduate class, you're like, oh. So, when I was in year 3 or 4 of my PhD, and occasionally I would feel stupid, right? I don't understand what these results mean. Am I stupid? Then I go teach undergraduate class, and I was like, oh, I actually know a lot about this sort of stuff. I'm not stupid. I actually know a lot. So that also helped me build my confidence, right?
Yeah. The act of teaching from the previous episodes, the act of teaching seemed to have also reinforced those concepts pretty deeply. Now, speaking of teaching, just quick shout out and link in the description, your wonderful R course that I'm actively following. What was the inspiration behind that? Because that is such a fantastic resource, and you put in so much time and effort into it.
Yeah, so there are a few things. So since, in my year 1 and 2, I spent hundreds of hours trying to teach myself R, right?
Mm-hmm.
And then one day I thought that it would be so nice if I can help other people learn, right? Because I already have all my notes and stuff. I can convert my notes into a course, and I can help other people learn. So you think that I spent a lot of time on it, but it's actually not as much time because I already have my notes, right?
Mm-hmm.
So that's one thing. If I can, it would be great if I can help other people learn, because I already did half of the work for them. That's one. And then the other thing is that in 2020, in spring 2020, well, when COVID hits, we all have to go home, right?
Mm-hmm.
But I still have undergraduate student who was going to work in the lab with me, and they need to do something so that they can get credit for their research for credit, right? So then I have to come up with stuff for them to do, and then the R course is that. So then I basically say, hey, here's this R course that you can do, and then in order for them to get credit, I have to come up with some sort of questions. Then I came up with some questions, and then I would just check their answers if they got it right. So that was the other thing. So those are two main things. that helped me, build those courses.
Yeah, that's yeah, thank you so much for that. That's such a useful tool, especially because like programming languages or whatever you want to call R, that weird, it is a language, it has programming. But it requires training, it requires practice, and I have taught and retaught myself R so many times, and yours has so far been the only one that's actually sticking. For some reason, I just like could not make R stick in my brain, right? Like especially like the ggplot sub layer of R. And yeah, no, please, thank you so much for that. That's fantastic. Now, with the remaining minutes, is there anything you'd like to plug or propose or advice that you'd like to give? The floor is yours, however you'd like to take it.
Yeah, so I guess I'll just, talk about the new additions to my GitHub repository. So in the past few months, so I had this quick, I think it's called quick data viz module about how to do graphs, and then it was listed under online R learning, but now it has its own GitHub repository. So if you could not find the quick data viz anymore, because now it has its own GitHub repository. And then I have made these new palette collections, color collections that I try to make colorblind friendly. And those are inspired by plants or pop culture. So if you're interested, you can find those. And then I also make a new tool for Symphony, vocal Symphony visualization using tidyverse and ggplot. And if you're interested, go check out that one as well.
Cool. Well, thank you so much, Lee, for your time. This was really fun, and I'm really looking forward to learning more from you. I really appreciated this.
Thank you.